All right, today we're jumping into one of the absolute cornerstones of modern biology, recombinant DNA. This is the engine that drives genetic engineering, and it has completely changed the game for medicine, for agriculture, you name it. So let's get right into it. I want you to think about something for a second. What if you could edit the basic instruction manual for any living thing? What if you could find a really useful piece of code in one organism and just paste it into another? Well, that's not science fiction anymore. That is the big idea behind recombinant DNA technology. So, first things first, let's break down what we're actually talking about here. What exactly is recombinant DNA? At its core, recombinant DNA, or rDNA for short, is basically a hybrid molecule. It's DNA that scientists have quite literally stitched together in a lab, taking genetic bits and pieces from different organisms. And the key thing to remember is that this is a deliberate, human-driven process. We're creating genetic combinations that you just never find out in nature. Now, here's a quick heads up, a little point of clarification that's super important. You might see the term rDNA and get mixed up. What we're talking about, recombinant DNA, is totally different from something called ribosomal DNA, which is a totally natural part of your cells. It's a small detail, I know, but it's one of those things you've got to get right to really understand the science. So why on earth would we go to all this trouble? Well, the whole point is to give an organism a brand new set of instructions. By popping in a new gene, we can essentially program a simple little organism, like a bacterium, to become a tiny factory. And that factory starts producing a very specific, very valuable substance, what we call a recombinant protein. Of course, to do this kind of incredible genetic surgery, you can't just, use your bare hands. You need a super specialized set of microscopic tools. So let's open up the molecular toolkit and see what's inside. It really boils down to three main players in this whole process. You've got enzymes, which do all the hands-on work with the DNA. You have vectors, which are basically the transportation system. And finally, you have host cells, which act as the production facilities. So let's start with the enzymes. You can just think of them as the molecular workforce. These are the proteins that are doing the actual cutting, the copying, and the pasting of the DNA. They're the ones getting their hands dirty, so to speak. Okay, so once you've got your new piece of genetic code all ready to go, you need a way to get it inside the host cell, right? That's what vectors are for. Think of them as the delivery trucks. Very often, these are tiny little circles of DNA from bacteria called plasmids. Their whole job is to carry that precious new DNA cargo to its final destination. And that brings us to the host cells. These are the factories. We're usually talking about simple, really fast-growing organisms like bacteria or yeast. Once that vector makes its delivery, the host cell reads the new DNA instructions and just starts pumping out the protein we want. And since it's also making copies of itself, you suddenly have millions of tiny factories all working on the same assembly line. All right, of all the amazing tools in this kit, there's one type of enzyme that is, without a doubt, the star of the show. We have to talk about the discovery that made all of this possible in the first place, the genetic scissors.
The official name is restriction enzymes, but honestly, molecular scissors is a way better description. Their function is just unbelievably precise. They don't just snip DNA wherever they feel like it. No, they recognize a very specific sequence of genetic letters and make a perfect cut right there. And it's that precision that gives scientists all the control.
And what's so cool is that this incredible tool wasn't invented, it was discovered. The story really starts back in the 60s when researchers noticed that bacteria had this weird ability to chop up the DNA of viruses that were trying to invade them. By 1970, the first of these enzymes was isolated, and in 1978, the pioneers who figured this all out, Arbour, Smith, and Nathans, were awarded the Nobel Prize, and they absolutely deserved it for this game-changing work. What's so fascinating is where these things even came from. They're not some newfangled human invention. They are, for all intents and purposes, a primitive immune system. Bacteria evolved these molecular scissors over billions of years as a defense mechanism against viruses, these things called bacteriophages. They'd just shred the viral DNA before it had a chance to hijack the cell. But hold on, that brings up a really obvious question, doesn't it? If a bacterium is just swimming with all these DNA-cutting scissors, what stops it from accidentally chopping up its own DNA? What stops it from committing genetic suicide? Well, the answer is just this beautifully elegant biological shield. Bacteria use another enzyme to add a tiny chemical tag, a methyl group, onto their own DNA, right at the spots where the scissors would normally cut. The restriction enzymes see this little do not cut sign and just move on, leaving the bacteria's own DNA completely safe. It's this brilliant system for telling self from other, and it leaves the invading unmarked viral DNA totally defenseless. And get this, these scissors don't all cut the same way. Some make a really clean, straight slice right through the DNA, which creates what we call blunt ends. But many others make a staggered cut, which leaves these little single-stranded bits hanging off. These are called sticky ends, and oh man, are they useful. Because they naturally want to find and pair up with a matching sticky end, they make the whole pasting part of this process way easier. Okay, so we've got the tools, we understand the basic mechanism, but what's the big deal? Why does any of this matter so much? Let's take a look at how this technology has literally changed our world. The impact wasn't some far-off theoretical thing. It was felt almost immediately. Just a few short years after the technology was figured out, a researcher named Herbert Boyer used it to put the human insulin gene into bacteria, and this was revolutionary. For the first time ever, we could make huge amounts of pure human insulin in a lab, which completely transformed life for millions of people with diabetes, who, before that, had to rely on insulin from pigs and cows. And insulin was just the beginning. This tech is the backbone of so much of modern medicine, producing hormones, blood clotting factors, developing new kinds of vaccines. In agriculture, it's used to make crops that can fight off pests or survive in tough climates. And for scientists in the lab, it's just a fundamental tool for figuring out what genes actually do. And just to give you an idea of the sheer scale of this toolkit, scientists have now identified over 3,000 different kinds of these restriction enzymes, and each and every one recognizes its own unique genetic sequence. This gives researchers this unbelievable library of super precise scissors to pick from, letting them cut and paste DNA with incredible accuracy. So this ability to rewrite the code of life, it has opened up a world of possibilities, right? It's helping us solve problems we once thought were just unsolvable. But it also brings up some really big, profound questions. The power of genetic copy and paste is immense, and it forces us all to think about not just what we can do, but where this incredible technology is going to take us next.